Hey young biologists, this is our first video for Unit 8 on DNA, the first of 3 units on how genetics work. Now, you've seen the DNA double helix structure in popular movies like Spider-Man, the picture shown here, but not a lot of people know the actual functions of DNA. Even fewer people know about the chemical structure of DNA, and even fewer than that understand how the structure and function of DNA fit together perfectly. Now, to welcome you into the club of awesome people who understand what DNA is all about, the topic of this video is DNA structure and function, and we'll tackle the essential question, what is the structure and function of DNA, and how do those fit together? Now, to connect back to what we already know, it's important to remember where exactly in cells DNA can be found. Remember, in eukaryotic cells like animal cells and plant cells, the DNA is found in the nucleus, right here and right here. Prokaryotes like bacteria still have DNA, but since bacteria don't have a nucleus, the DNA will float around in the cytoplasm. So a key message here is that all living things have DNA, and the DNA of eukaryotes is in the nucleus. As seen in the movies, DNA is twisted up into this double helix that you can see here and you can see here. A double helix. Now, twisting up the DNA helps compact the DNA down, but did you know that the DNA from a single human cell is 6 feet long? So you need to fit 6 feet of DNA into a teeny tiny nucleus that's smaller than the smallest dot you can make with your pencil. So DNA is twisted into a double helix, this spiral staircase shape, but then the double helix is twisted and packed even more into structures called chromosomes. So DNA is twisted into a double helix shape and then tightly packed into chromosomes so that the 6-foot-long DNA molecule can fit into the teeny tiny nucleus. Now, if we zoom in to that double helix, we can see the individual pieces of DNA. Now, the name DNA tells us a lot about the chemical structure of DNA. DNA stands for Deoxyribo nucleic acid. DNA is a polymer made up of monomers. Polymer means many pieces, and if you look closely at DNA, you see a pattern of all of those pieces. Each of these pieces is a monomer, and when the monomers link together, you get a big DNA polymer. If we zoom in closer still, we can see the parts of each monomer. So here's one monomer. The monomer piece of DNA is called a nucleotide, and each nucleotide has 3 parts. In the middle of it all is the sugar. And in DNA, this sugar is called deoxyribose. In fact, that's what the letter D stands for in DNA. So this pentagon is the deoxyribose sugar. Connected to one point of the sugar is this other chemical group called a phosphate group, shown as this circle right here. And then connected to the other side of the sugar is the base, shown as this shape right here. So each nucleotide monomer is made of a phosphate, a sugar, and a base. Another key thing to know about the DNA monomers is that all of the monomers have the same type of sugar and phosphate group, but there are 4 different possibilities for the base. Those possibilities are thymine, adenine, cytosine, and guanine, or T, A, C, and G. To make sure you know that these are different, I've just drawn them as 4 different shapes here. So to turn those nucleotide monomers into a DNA polymer, first thing we have to do is line up a bunch of nucleotide monomers. Now we've got to link together those monomers. To join the monomers together, the phosphate of one nucleotide is linked to the deoxyribose sugar of the next nucleotide monomer. Here's one of those chemical bonds, and here's another of those chemical bonds linking the monomers together. It's really important that these DNA strands don't fall apart, so the bonds between the monomers have to be strong. In other words, these bonds need to be our strong, covalent bonds that are like a firm handshake between different chemicals. But remember, DNA is a double helix. Now, the double means two strands of DNA, one strand of DNA, and the second. So, if one strand is joined together with covalent bonds between the nucleotides, how do we get those two strands to join together? Well, a diagram like this one here will help us understand better. Here are the two DNA strands. Notice that the focus is more on the bases rather than the sugar and the phosphates. And if you take a close look, you should start to notice some patterns. First, the base parts of the DNA are what line up. So, in a double helix, the bases of one strand join with the bases of another strand. In addition, the bases match up in a specific way. You'll notice that A is always opposite of T, here, here, and here. And C is always opposite of G, here and here. Now, this matching is referred to as the strands being complementary. Complementary simply means that the strands match up or fit together in a certain way. The rule is that A is complementary to T and C is complementary to G. Now, the bonds that hold bases together to make the DNA double-stranded are hydrogen bonds. And these are the same bonds we met when dealing with water cohesion and water adhesion. Remember, hydrogen bonds are weak bonds, like tiny little finger bumps between molecules, and having weak bonds holding together the double helix lets the double helix get unzipped, so DNA can be used for its job. Now, this obviously brings us to the question, what exactly is the job of DNA? For most people, DNA is the molecule that makes your hair black and your eyes brown, or what makes you more or less likely to get certain diseases. But the actual job of DNA is a mystery for most people. And with that in mind, I tried to get the weirdest, most mysterious picture of DNA. And I think I succeeded. Yeesh, look at this guy. What is DNA exactly doing? But the truth is that the job of DNA is pretty simple. First, DNA needs to replicate itself. Because DNA can copy itself, each new cell that's made gets an exact copy of the DNA. And second, DNA stores information. The information stored in DNA is like a big recipe book for all of the proteins that are needed to make up each cell in your body. Now, hopefully you can put things together to understand how each strand is put together with strong covalent bonds, so this information doesn't get messed up or mixed up in different order, but the strands are linked with weak hydrogen bonds so the information can be accessible by a simple unzipping. So this was a big but important introduction to what DNA is all about. So let's recap some of the major points. First, DNA is a polymer made up of nucleotide monomer pieces. In order to fit into a nucleus, the DNA strands are first twisted into a double helix to make them a little more compact, and then compacted even more into chromosomes so that you can get 6 feet of DNA into a teeny tiny nucleus. Each nucleotide is made up of the same sugar and the same phosphate, and the third part of the nucleotide is going to be one of 4 bases: adenine, thymine, cytosine, or guanine. These nucleotide monomers are strongly linked together to make a DNA polymer using covalent bonds, strong bonds that keep the DNA strands from falling apart. But 2 DNA strands are joined together by weak hydrogen bonds. The weak hydrogen bonds allow the DNA double helix to unzip and allow the cell to access the information stored in the bases A, T, C, and G. Finally, the job of DNA is an instruction manual for proteins And that instruction manual is copyable so that each cell and each new cell that's made get a copy. Hopefully this is a good introduction to DNA and is going to set the table for our unit about DNA.